Hello, my name is Lauren Estes. I'm an RA in the Department of Psychology under Dr. Helen Sable. And today I will be presenting working memory impairments in an animal model of Attention-Deficit Hyperactivity Disorder, the SHR rat. So SHR is also known as Spontaneously Hypertensive rat. It's a model of ADHD that has been examined by a lot of other groups, and it's a relatively well-accepted animal model of ADHD. This project is just part of a larger one comparing SHR to other prospective ADHD models. In particular, this project focuses on the working memory of the SHR models in comparison to the behavior of other prospective models. The controversy about working memory deficits as a core symptom of ADHD, one is that some people believe it underlies impulsivity, while other people believe that one can occur without the other. So here we have our subjects, SHR and WKY. WKY is the control. Both groups had 24 subjects in it, 12 male and 12 female. Food restrictions started at postnatal day 60, while testing began at postnatal day 70. The food restriction was 85% of their free feeding weight with biweekly adjustments for growth. And this was done to keep them motivated to respond for food within the trials. These are the apparatus. There were 10 in the lab, and each one had 2 levers with a cue light above each lever. In DRL, we only used the right lever, but in all others, the left and right were both used. For each successful trial, a 45-gram food pellet was dispensed into the food magazine in the middle. In the procedure, all phases used a strict alteration procedure in which the correct lever on a given trial was opposite from the lever previous where the rat responded. And all of the rats were previously trained to lever press for food and on an inhibitory control task. So first we have cued alternation, also known as CA, where the rat has to press where the cue light is illuminated over the lever, and the cue light alternates from one side to the other until they achieve 60% correct. Next, we have non-cued alternation, which is basically the same as CA, but there is no cue light to show. And then we have DSA, which is the same as NCA, but there are delays between the trials that differ from 0 seconds, 5 seconds, 10 seconds, or 20 seconds. And there are 25 sessions of those. So a little bit more about DSA. The key here is that the rats must keep in mind where they responded last in order to know where to respond on the next trial. So here we see that the rat would press the lever on the right and be rewarded with a food enforcer. Next, there would be a 0, 5, 10, or 20-second delay. And if the rat were to press the lever on the left, they would get another food reinforcer. But if the rat were to press the lever on the right, they would not be rewarded. There would be no food reinforcer. They must alternate from the left to the right or from the previous to the next in order to get a reinforcer. And of course, as the delay gets longer, the task just gets harder. So here's a video of a different strain of rat conducting a test like this. If the rat fails to alternate, you'll see it gets no reinforcer. There is no music in the lab. This was just for our enjoyment. But so next we have our data analysis of the NCA test. The percent correct is the dependent variable, with strain, sex, and the day being the independent variables. The analysis will be a mixed ANOVA, a 2x2x10, and the analysis for this will be mixed because sex and strain are between subject factors, while the day is a repeated measure factor. So similar, we have DSA, where the percent correct is the dependent variable, and the data across 25 days will be averaged into 5-day testing blocks. And then we have strain, sex, and testing block as the independent variables. And again, we have a mixed ANOVA test, a 2x2x5. So NCA, our anticipated results, is that there is no working memory impairment, so performance of the SHR and the WKY, the control rats, should be fairly similar. No difference on NCA also allows us to rule out a potential motor impairment, so if we do see a difference on DSA. So NCA works as a good control task as well. Our DSA anticipated results are that the percent correct will decrease as the delay gets longer. As I said earlier, as time goes on between the trials, it does get harder for the rats to pick the correct lever. SHR rats, we believe, will exhibit a lower percent correct at shorter delays when compared to the control rats. We don't expect to see a sex difference, although we will look for it and report it if there is one. And the take-home message on this is that our results will indicate whether a non-animal model of ADHD, the SHR rat, does indeed exhibit working memory deficits. And these results will be combined with other results from our lab examining impulsivity in these rats to provide more information about these behaviors in ADHD that can then be applied further into our everyday life. A special thank you and kudos to Dr. Helen Sable, Joshua Potter, Nick Page, and the other RAs in the lab. Thank you.